peptide bonds. What else? Only peptide bonds are found in proteins?
No, ma'am. Hydrogen bonds and salt bridge
Hydrogen bonds.
covalent bonds.
Covalent bonds,
bonds.
salt bridge, hydrogen bonds. Very
Sulfide
good.
bonds.
Acha, nice. Here people are studying. Tell me two acidic amino acid.
Ma'am, aspartic acid and glutamic acid.
Aspargine and glutamine.
Oh, sorry.
Aspargine,
Aspartic
glutamine,
acid and
aspartic
glutamic
acid.
acid.
Say, okay, good. And what about some basic amino acid?
Arginine, ma'am.
Arginine and
Arginine and lysine.
lysine. Okay, good. Good, very good. And what are polar amino acids?
Threonine and cysteine, asparagine, glutamine.
Okay. So serine, threonine, they have the OH group, hydroxyl group, they are polar in nature. Okay, so you have done good studies in for proteins. Chalo. So we have little few things left for protein, and then we can go over to nucleic acids. So can you see my screen?
Yes, ma'am.
Okay,
Yes, ma'am.
so proteins hierarchy we had discussed in the last class. Proteins are of different in different classes of proteins are there. The primary structure, then they fold into with the help of hydrogen bonds. They form the secondary structures, which consist of alpha helices and beta sheets. And then we move to tertiary structures when more of the secondary structures come together to form the tertiary structures. And when many tertiary structures or many polypeptide units join together to form the quaternary structures. So this is the hierarchy of the proteins. So proteins can be of very varied shape and sizes. So as you can see in this figure, you can see then different shapes of proteins are there. Some are small, some are big. You can see the structure of the hemoglobin protein that is found in your bloods. And then you can see the collagen protein, which is a triple helix kind of thing. Then myoglobin, and then many more, catalysis and enzyme. Then you have other structures over here, which are insulin and all. So proteins, different forms. Okay, so one more important thing when we are discussing about protein is, I'm not going to focus much into this, but you should be aware of this. What is these things? Because you all have seen this, how we, while making an omelette or while warming a milk, what is the changes that is happening. That are the things that you have seen in your daily to like in your kitchen, right? So what is actually happening is that proteins usually have a three-dimensional structures, stable structures, but with the help of certain agents which can be external or internal, like it can be heat, it can be changes in the pH or certain detergents like urea or something, they can denature the three-dimensional structure of the proteins. And once that structures get disrupted, that function is lost. So that structure to let that protein denaturation can be of like permanently denatured, or it can be temporarily denatured. Okay, for example, when we are doing a making an omelette, this is kind of the structure is completely denatured. But when we are warming a milk, this is a kind of the structure is temporarily denatured. So it can be again get back to its old form. So protein denaturation, why I am discussing this is because you look into, there are certain proteins which are known as prion proteins. Prion proteins, what they do, they have this ability to change the structure of proteins, and that is because of that there are many neurodegenerative diseases. Okay, so prion proteins and all that. So many degenerated proteins are very important part of life. But again, then the structure, when they get denatured, they lose their function, and that is why many diseases happen. So this was all about proteins, I guess. So we have summarizing.
So what is prion?
Prions are a type of proteins. Okay, prions are a type of proteins. I mean, this kind of misfolded proteins. So they have this ability to change the other proteins. Okay.
Ma'am.
Yes. Yes.
Ma'am, when we boil an egg, the egg yolk gets denatured. So, ma'am, can you please explain that why the egg yolk means get denatured in the protein?
There are the hydrogen bonds, etc. No, the structures that gets changes. So when we are heating, the heat actually breaks the bonds and all. Okay. That is
Okay.
like a three-dimensional structure that gets disrupted. Okay, suppose we are like having a three-dimensional structures which are having certain hydrogen bonds, hydrophobic interactions and all that. Okay, so when we are putting some external agents and all that, so what is happening is that disrupting that bonds within that structure, and that is why that entire structure gets changed.
So, ma'am, it solidifies because of the means boiling of the water inside that, or means some chemical reactions are being formed and so that it solidifies.
The heat is actually putting the effect. No, the hydrogen bonds are breaking.
Okay. Okay, ma'am.
Heat is the agent over here, which is actually putting effect on the hydrogen bonds. Yeah, so these are the summary point. Amino acid, you know, proteins are form of amino acids. Denaturing, I discussed because the structure has changed. Peptide bond, you know, they are between the different amino acids to form the proteins, and then polypeptides, the chain of amino acids linked by the peptide bonds. So that is all about proteins. We are moving to the fourth macromolecule. We have already discussed about carbohydrates, lipids and proteins, and the last is nucleic acids. And today we'll be finishing nucleic acids, and with that we are finishing our biochemistry module. Okay. Nucleic acids, when we talk about, there are actually basically two nucleic acids, the DNA and the RNA. DNA, the full form is deoxyribonucleic acid, and RNA, ribonucleic acids. So these are basically the genetic material, okay, which is actually present in all organisms. Mostly in all higher organisms, everything, DNA is there, and only in cases of viruses, the RNA is the genetic material. The basic difference you can see over here, the structure of the DNA and RNA. What you can see, this is DNA and this is RNA. With the in terms of structure, can you tell me what is the basic difference of these two structures?
Double-stranded and single-stranded.
Yes, double stranded means there are two strands wind together in the DNA and RNA. There is just one strand. Okay, that is the basic structural difference. There are many other differences. We'll be discussing them soon.
And one more is there. The uracil base is only in RNA. It is replaced by the.
I know, there are many more differences. We'll discuss. I'm just talking about what is the visually you can see this two structure. What you can see. This is something double-headed and this is single. Right? That is what I was talking about. Yeah, there are many differences. We'll discuss. Okay. Now coming to the monomers of nucleic acid. Sorry?
This part will be not coming in the CA1.
No, this is not coming.
Okay.
Whatever I had taught in that last class till protein, that is there.
Okay.
Okay. So what are the monomers of nucleic acid? Any guesses?
Nucleotide.
Nucleotide.
Nucleoside.
Nucleoside. Getting confused. What is nucleotide? What is
Nucleotide, ma'am.
Tide ya side?
Nucleotide.
Ma'am,
Nucleotide.
nucleotide is the thing which has the base, but nucleoside doesn't have, I think.
Oh, like that. Oh, nice. Okay. So nucleotides are the monomers of nucleic acid. Don't get confused in the exam. यहाँ पे TID के जगह SIDE लिखोगे तो अंडा मिलेगा, zero मिलेगा. Okay. So nucleotides are the monomers of nucleic acids. So many nucleotides, they join together to form a nucleic acid. And how does a nucleotide looks like? So this is the structure of a nucleotide. You can see there are three components. So this orange one, this is say sugar. I am showing here a ribose sugar. And then it is attached to a nitrogenous base. Here we have a nitrogen base known as adenine. There are many nitrogenous bases. We'll be discussing them also. And then there is a phosphate group over here. There are three phosphate groups linked together here. So this three totally composing a form a nucleotide. And then When this type of nucleotides, many nucleotides come together, they bind each other, they form a nucleic acid, which is a macromolecule. So this is the basic structure of nucleic acid. As I said, one nitrogenous base, 5 carbon sugar, and then is one or more phosphate group. Okay. So there are, it is color coded. Base is shown in green, phosphate in yellow, and sugar in blue. Now someone was talking about nucleoside. Let's see the structure of nucleoside. How does they look like? So this is the common difference between nucleotide and nucleotide. So the phosphate is missing in nucleoside, and that is why that is the major difference between nucleoside and nucleotide. So someone was talking about what, sugar and phosphate, right? So sugar and phosphate nahi hai, yahan pe base और sugar hai in nucleoside. So phosphate is missing in nucleoside. When that phosphate group is added, see one P over here is added, so it forms a nucleotide. This sub nitrogenous base, the blue one, sugar in green, and then phosphate in violet. Color coded. I have given the color coded. I think this is the basic structure. I think it won't be difficult. It will be a difficult problem for you to remember this structure also. Isn't it, class? The basic framework, structural framework of this nucleotide and nucleoside. Sugar, base, phosphate in nucleotide, and simply base and sugar in nucleoside. Okay. So nucleotides actually have different functions. First of all, they are the subunits or the monomers of DNAs and RNAs. So that is why they are the repositories of the genetic information. The genetic information is present in the nucleotides, which are actually forming the nucleic acids, and we call the DNA and RNA to be the genetic material. Genetic material in the sense that you have been seen that you have been given many compliments like, okay, your eyes looks like your father, your nose is like your mother, and so on. Your hair comes like your father, and on. So what is this all? Because these are the genetic, the DNAs get transmitted from the one generation to the next, from the parents to the offsprings. So that is why there are resemblance in the many resemblance in the of the child with the parent. This is all about genetic material. Okay. Nucleotides have other functions like they are energy carriers. You can see, looking at the ATPs, which are actually released in different cycles. We have learned about TCA cycle and the glycolysis and all. Then they are also enzyme helpers like acid coenzyme A, you can see. And then they are chemical messengers also to help in signaling, cell signaling, which you will be learning in cell biology. Hopefully, Munirpa Ghosh ma'am is teaching you that cell biology. You will be learning that about cell signaling. So this way, nucleotides have many different functions. So yeah, I was discussing about the 3 components of nucleotides. So let's discuss in a more detailed manner. So there are 3 components, as I said. One is nitrogenous base, one is sugar, and one is phosphate. So for the nucleotides, usually there are 5 nitrogenous bases. These nitrogenous bases can be of 2 categories. One is purine, one is pyrimidine. Just look into this structure wherever I'm putting my pointers. You see the structure of purine and see the structure of pyrimidine. What is the basic difference you can see over here?
Rings, number of
Double
rings.
ring, single ring.
Yeah, the number of the rings. You can see in the purines there are 2 rings, and pyrimidine there is a single ring, benzene ring. So that is the major difference between these 2 bases. So purines have actually 2 rings, and usually there are 2 purines, adenine and guanine, and pyrimidines there are 3, thymine, cytosine, and uracil. So I have seen students who make mistakes, like now we are talking about glycine, glycerol. So don't get confused with cytosine and cysteine. Okay, guanine with something else, glycine and all, glutamine and all. So adenine, guanine, thymine, cytosine, uracil. 5 nitrogenous bases are there. Yeah.
Ma'am, we don't need to remember the exact structure now for these.
Of these things.
Yes, ma'am. For
Yeah,
these
don't have to remember the entire structure, but you can just see the framework. That is the 6 ring with the 5 rings with the of the purines and the only one single ring of this pyrimidine. At least this much you should be knowing. Okay. But I've seen students who actually make the structures also very nicely in the exams. Okay, so that's up to you. Yeah, what were we discussing about? These are 5 different nitrogen bases that are present in the nucleotides. Now coming to the sugar part, the sugar can be of 2 different sugar. One is deoxyribose, one is ribose. Now again you see the structure of this 2 sugar. What is the difference?
Oxygen.
In the second part.
Oxygen
And
is
carbon
2 number carbon पे oxygen गायब है.
is there.
गायब है. गायब है. Okay, good. Second carbon, if you can see, second carbon in this sugar molecules in ribose have a oxygen while it is missing in the deoxyribose. So that is why if you look in the chemical formula, in ribose we have the formula C5H10O5. When is deoxyribose, we have C5H10O4. One oxygen is missing. Okay, in deoxyribose. So you can see in the DNA we have the deoxyribose sugar, in the RNA we have the ribose sugar, and hence the name ribonucleic acid and deoxyribonucleic acid based on the name of the sugar molecule, because sugar is the main thing that is actually main difference between the 2 DNA and RNA, because of which we are having the structural differences as well. We'll discuss that. Oxygen in deoxyribose is missing in the second carbon atom. Now looking into the phosphates are actually normally joined to the which carbon? The 5-des hydroxyl of the sugar, ribose or the deoxyribose. So this carbon atom you can see, we can numbering of carbon you can see 1, 2, 3, 4 and this is 5. So this is 5th carbon atom. To this the phosphate actually get connected, joined. And then phosphate can be of monophosphate, diphosphate or triphosphate. Mono means 1, di means 2 and tri is 3 that you know. So this is how 1 phosphate group, 2 phosphate group and 3 phosphate groups. And this phosphate molecule actually makes this nucleotide a negatively charged molecule. Okay, because of this phosphate groups, nucleotides are negatively charged in nature. See how the phosphates are linked and what is the bonds in nucleic acids. So we have talked about the bonds in carbohydrates. What is the bonds in carbohydrates? Class?
Glycosidic bond.
Glycosidic bonds. In fats it is fascinating. In proteins it is
Peptide bond.
Peptide bonds.
And in nucleic acid we are having this phosphodiester bond. Okay, phosphodiester bond. Now if you look into this, so I have shown you here 1 nucleotide, nitrogenous base, 1 sugar and 1 phosphate molecule. Similarly, this is another nucleotide with a sugar with nitrogen base and 1 phosphate molecule. So what is the difference of this two, first nucleotide and second nucleotide structurally? Check. Are they similar nucleotides?
No, ma'am. In one
Bases
there
are
is
different.
purine and in other there is pyrimidine.
Yes.
Bases are different.
Bases
Yeah,
are
that is-
different.
Here we have a pyrimidine base. Okay, this is 1 ring. Here is a purine. They have 2 rings. Okay, these 2 nucleotides. Good, very good class. And then what we are seeing, how this phosphodiester bonds are being formed. You can see that the 5th carbon of this nucleotide 2 is having a phosphate group over here. So what happens, this phosphate group that is attached to the 5th carbon of this nucleotide 2 interacts or joins with the hydroxyl group of the 3rd carbon of the nucleotide 1. Okay, 5th carbon of one nucleotide has a phosphate group and 3rd carbon of the 2nd nucleotide has a hydroxyl group. So there will be another condensation reaction. What has happened in condensation reactions? 1 water molecule is released. So this phosphate and this hydroxyl group, they form together to form a phosphodiester bond with the releasing of 1 water molecule. Reaction easy here. 1 water molecule is being removed. Class?
Yes, ma'am.
Okay. So this OH with this
Yes, ma'am.
hydrogen gets removed and we have a double bond. We have a bond between this phosphate with this oxygen of this 3 des carbon. Clear? So DNA and RNA, these nucleic acids, are covalently linked together. Covalent bonds, covalently linked together with the phosphate groups of the fifth phosphate group of one nucleotide, which is joined with the 3-hydroxyl of the second nucleotide, forming a phosphodiester linkage or phosphodiester bond. So this way, simultaneously, many nucleotides join together to form a bigger molecule known as nucleic acid. So this is how a phosphodiester bond. Phosphodiester bond you can draw. I don't think it will be a difficult thing. Class?
Yes, ma'am.
Peptide bond, phosphodiester bond, सबका same ही है, बस water removal है. It's not that difficult. Okay. Yeah. So phosphodiester bonds have certain properties. Because of the phosphodiester bond, what is happening? See. So first of all, nucleic acid, the backbone is hydrophilic in nature. What is happening? The hydroxyl group of the sugar. So we have a sugar molecule over there, the blue ones representing. So what is happening? These hydroxyl groups are actually form the hydrogen bonds with the water molecule. So that is why this backbone of the nucleic acid is hydrophilic in nature. They are polar. They love water. And then these phosphate groups are completely ionized and negatively charged at the pH of 7. And the negative charges are generally neutralized by the ionic interactions with the positive charges on the proteins, metaions, etc. So what is happening? Since this negatively charged nucleic acids, they interact with the positively charged, let's say proteins or let's say metals, ions, and in the cellular environment, and that is why we get a stable structures. So what is happening? We have this DNAs, RNAs moving, and then they form bonds with the protein molecules, and then they form some stable structures. Okay, so and because since they are negatively charged nucleic acids, they attract the positively charged of the protein molecules. Now this is something I don't know whether you have learned or not. I guess those who have biology must have learned, but I don't know whether the non-biology students have heard about it or not. So this is a famous. The structure of the DNA was given by this famous scientist James Watson and Francis Crick, because of which they got this Nobel Prize. And this was all possible due to the X-ray crystallography. And there are other people like Rosalind Franklin and Maurice Wilkins who actually contributed to this, so that these people could deduce the structure of the DNA, which is a double helical structures. So they had no idea that how does a DNA looks like. So we are reading about DNA is a double helical structure, but the structure actually was introduced by these two people with the help of X-ray crystallography. So based on this double helical model, how does the DNA looks like? As I said, there are actually two strings. This is one chain, nucleic acid chain that I told, sugar base phosphate linked to phosphodiester bond. So two strands, when they fold upon each other and they form a double helical structures. So there are certain features of this Watson-Crick's model which shows that actually there are, when there is one rotation, one turn, complete turn, there are actually 10 base pairs. 10 base pairs. We will discuss about what are base pairs. These are the bases in each strands. They form. They join together with the help of hydrogen bonds, and in one turn there are actually 10 base pairs, and this height will be around 36 Armstrong. But if you divide this 36 Armstrong divided by 10, so this is actually a little, this should ideally come 3.6, but it is not 3.6. The actual distance between two base pairs is 3.4. So there is a little change. I mean, not exactly 36, 10 base pairs with the 36 Armstrong is moving. So that is why it is around, some books write 3.4, some write 3.6. So not a big issue. You can write anything, 3.6 or 3.4, that is okay. The turn, the distance. And because of this turning of this DNA, so there are groups created, which are major groups and minor groups. Which is major group, you can see this one is a major group, in the sense that it is more narrow, and this more more wider, and this is minor is more narrow kind of things. Okay, so let's talk about the DNA double helical structure in detail. So if you can just unwind this double helical structure, you can see that it looks like a ladder. Okay, if I am just removing the strand, that is unwinding the strand, which is actually wound upon each other, so I can see that this is forming a ladder like structures. So the legs of the ladders, this is the main strand, this is the two. They are actually consist of the phosphate and the sugar backbone. I was discussing about the phosphodester bond. So phosphate and sugar are actually forming these two strands, while this middle ones, the rungs of the ladder, the joining part, are actually forming the nitrogenous bases. Okay, so the nitrogenous bases form this middle part, and the two strands of this ladder, they are composed of the phosphate and the sugar backbone. This again is structure of these things, which in the form of common chemical. So this is phosphate groups, and this is the sugar, and here the bases. So this is opened up. You can see this is one strand, and this is another strand. They are joining together with the base pairs. So you can see this is a what kind of molecule? Pyrimidine, and this is a purine. So the purine and pyrimidine are interacting with each other. They are actually forming the middle part of the ladder of the helical structure, and this is are forming the legs, the phosphate and the sugar. So now when this base pairing happens, there are certain rules for base pairing in the DNA as well as the RNA. So that is known as the Chargaff's rule. The Chargaff actually proposed this rule, and he found that purines are actually forming bonds with the pyrimidines, but again purines consist of like adenine, which is a purine. It is binding with thymine, which is a pyrimidine. So A is pairing with T. Similarly, guanine is pairing with cytosine. So this is actually the rule that is followed in the DNA while they are forming the double helical structures. So adenine, which is shortly written as A, and thymine is written as T, guanine written as G, and cytosine as C. So ATGC, AT and GC. AT has a bond. This is a double bond between A and T, while in GC there are triple bonds, triple hydrogen bonds. You can see how the structure is over here. So AT are binding together with the help of hydrogen bonds, and GC with triple hydrogen bonds. And in case of RNA, we have uracil instead of thymine, so we have adenine pairing with uracil, AU. Okay, GC is same in both DNA and RNA. GC base pairing is same in both DNA and RNA. In case of DNA, it is pairing A with T. In case of RNA, it is A with U. So suppose you know the amount of, let's say in a DNA, I can say there are 40% thymine. Can you tell me how much adenine is there?
40%.
40%.
40%.
Same, ma'am. Same.
Complementary.
Yeah.
Because
And now you can tell about guanine and cytosine also, how much they are?
60.
Each 10%.
60%. Okay.
10%.
Same percent. So A and D, A will T, how much A is there? Same amount of T will be there. This is the structures again. I'm talking about double helical structure, adenine and guanine, and thymine and cytosine, 4 bases in DNA. You can see how this base pairing, G and C triple bonds. You can see 3 bonds, and A and T there are 2 hydrogen bonds. AT 2 and GC 3. And this is the sugar and phosphate backbone. Let me see here one thing. Can you see one thing, that how this is this structure is in the sense that, can you see the polarity? One strand is pointing from 3' to 5', and another strand is pointing from 5' to 3'. Okay, this is known as the antiparallel bonding in the DNA. So first of all, complementary base pairs This is complementary base pair in the sense AT and GC are binding to each other. And again, these two strands are anti-parallel in nature there in the sense that one is moving from the 5' end to the 3' and another is moving from the 5' to 3' to 5' and all that. Okay. So this is another feature of the DNA double helical structures, that the two strands do not move into the same direction. They are moving into the opposite direction and they have this base pairing thing which are following the Chargraph's rule, AT and GC, with the 2 hydrogen bonds in AT and 3 hydrogen bonds in GC. And the sugar and phosphate background are there. Yeah.
Ma'am, what is the meaning of this 3' and 5' actually?
3' and 5' actually, if you look, okay, I'm just going back to this structure, you can see. Okay, so what is happening over here? You can see this thing, 3' to 5' thing, in the sense that this end, the 5' molecule, 5' carbon is attached to this phosphate group. So if you can look, this is moving into this direction on the 3' to 5' end. In the sense the 3' is at the end and 5' is moving up. Okay, so if this, and just if you rotate this one in the opposite way, so what will happen? This entire thing, that is this sugar is pointing upwards. You can see this pyramid structure. If you can just rotate it in the other way, it will be the other way. 3' to 5' at the end way, other way. So 3' and 5' is actually the sugar notation. Okay, how the sugar is there. See, you can see this 3' end, and this is the 5' end. Okay, you can see this way. Just you can see in this structure also how these rings are moving. This sugar is on the top upper side, 3' to 5'. Here it is on the lower side. You can see the oxygen is pointing down. Yeah.
Means sugar, 3 number carbon of sugar is 3 number carbon of sugar is lower the 3' and 5 number carbon of sugar is upper the 5'.
Yeah, exactly. Based on the sugar notations, you can see the structure, you know, here. ये कैसे हुआ है? Sugar का structure point to upwards हो रहा है. This 3' to 5' end, and here it is pointing downwards. The sugar. Can you see? Clear, class?
Yes, ma'am.
Okay.
Yes.
Good.
Yes, ma'am.
This is about the DNA structures, and you can see, you can find, read more about the Watson-Crick model. This is a very important question. Maybe in your CA2 you can get that question. I'm telling you again and again. Read about the DNA double helical model, helical structure. What are the salient features of this Watson-Crick model, which is a probable question which usually comes in exam. So I'm just telling you to read about that. Now moving to the ribonucleic acid or the RNA, you can see this is DNA which is double helical and RNA is a single helical structure. So what is happening? RNA is actually formed from the DNA, and the phenomenon is known as transcription, which we'll be discussing more about in the molecular biology class. So DNA to RNA, that step is known as transcription. I had told about another term when proteins are formed from RNA to protein. Does anyone?
Translation.
Okay. Okay. Very. So I think those who are answering must have biology in their class 12th. So that's fine. So in molecular biology, we'll be discussing about these two things, transcription and translation. Yeah.
I didn't have biology.
Oh, very nice. Good. Who is this? Harsh.
Yes, ma'am.
Okay, good. Nice. So transcription is the step from DNA to RNA, and RNA to protein. We have translation. So what is happening here? So the major difference between the DNA and RNA, as you can see, DNA is two strands, double helical structure, but in RNA we have just one single stranded. And the basic another difference that we had discussed, the sugar. In DNA we have deoxyribose, and in RNA we have deoxyribose and ribose. And then instead of the nitrogenous bases I had discussed, again I had also told that part also. So in DNA we have this nitrogenous base known as thymine, which is represented in T, but in RNA we have uracil and no thymine. So uracil is replacing thymine in RNA. Okay, so DNA and RNA structures. I have listed some key differences for you. So DNA is deoxyribonucleic acid, RNA is ribonucleic acid. DNA consists of 2 strands arranged in a double helical manner, but RNA has just 1 strand in terms of structure. Okay, in terms of sugar, we discussed DNA has deoxyribose, RNA has ribose. So deoxyribose has 1 oxygen less in the second carbon atom. And bases, the bases in DNA are ATGC, adenine, guanine, cytosine, and thymine. In RNA, A, G, and C are same as DNA, but uracil is replacing thymine. Okay, so similarly, the base pairing, if you can look like, so A, T, and GC base pairing is happening in DNA, while in case of RNA, A is pairing with uracil, so AU and GC. Location where DNA and RNA are found. So DNA is actually found in the nucleus, and also some amount of DNA are present in the mitochondria as well. But in case of RNA, RNA forms in the nucleolus, then it moves to specialized regions of the cytoplasm depending on the type of RNA formed. I think in cell biology you must have read about different types of RNA. Did you read or not? Transfer RNA, messenger RNA, ribosomal RNA, little bit
Yes,
of.
ma'am.
Okay, so RNA has different forms. So based on the type of RNA, so they are kept in different regions. Now moving to the function, what is the function of DNA and RNA? So DNA is really the genetic material. It actually replicates and forms many copies of, like, the DNA replicates and forms DNA again. So this is a blueprint of all the genetic information that is present in any organism. What is actually happening in RNA is that it is actually converting that genetic information of the DNA, and it is forming proteins, which is actually the building blocks in any organisms. So what is actually actually bringing that information from the DNA and passing on to the proteins, right? RNA is the intermediate one, or the messenger one, you can see. Okay, so that is the key differences between DNA and RNA. So let's summarize what we have discussed. So we know about ATP. ATP is a nucleotide that consists of 3 parts, one a base, one sugar, and one phosphate, 3 phosphate. And the name says adenosine triphosphate. Adenosine means what kind of base it is having? Nitrogenous base. It is having adenine. Adenine is the nitrogenous base. Tri or 3 phosphate group, 3 phosphate groups are there. So adenosine. So we are discussing sugar, and then sugar is which sugar it is containing. So there is a ribose sugar, adenosine triphosphate, and there are 3 phosphate groups. So this function as an energy carrier. DNA we discussed, this is the genetic material which consists of 2 nucleotide chains which are twisted in a double helical manner. Nucleic acids are many nucleotides which are joined together by the phosphodiester bonds, and then nucleotide are the monomers of nucleic acids. They have 5 carbon sugar, which can be deoxyribose or ribose, and it contains 1 nitrogen containing base, which can be purine or pyrimidine. Purine consists of A and T, and pyrimidine consists of
G and C.
CG.
यही तो गलती हो गया, मैंने बोला तुम लोग सुन लिए, जीसी है pyrimidine?
नहीं मैम.
क्या है बताओ?
Cytosine, thymine, and uracil.
Thymine and cytosine.
And uracil also. See, this is how happens. I am saying in something flow. You are not interrupting. मैम आपने गलत बोला. मैंने बोल दिया AT है. AT कहाँ है? Purine. A and G, adenine and guanine are the purines, isn't it? AT तो base pairing होता है. A is for purine, T is for thymine. Thymine is pyrimidine. A is purine. ठीक है? Prions are infectious proteins. I don't know why I put it here. Anyways, okay. RNAs are single-stranded nucleic acid which has role in protein synthesis. So that's it. So here I have put one cartoon form for you. Carbs, lipids, protein and nucleic acid we discussed. So you can see what are the elementary composition. So carbon, hydrogen, oxygen are present in all the three macromolecules. You can see CHO, 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 CHO. In protein, CHON, and then there is one sulfur also. So CHON, and in nucleic acid, CHONP. So nitrogen and phosphorus are also there in nucleic acid, the last one. But if you saw elementary composition, carbs and lipids are the same elements: carbon, hydrogen, oxygen, carbon, hydrogen, oxygen. Proteins have carbon, hydrogen, oxygen. Also nitrogen is there, and some sulfur is there because cysteine contains sulfur. And then in nucleic acid we have carbon, hydrogen, oxygen, nitrogen as well as phosphorus. Clear class? That's it. We are done with biochemistry module today. And if you all have any doubts, please let me know. So biochemistry, I think it was not very difficult for you people because this is more of chemistry rather than bio. The basic we discussed about 4 macromolecules: carbohydrates, proteins, lipids and nucleic acids. Their monomers we discussed. What are the monomers? What are the bonds between these things? What are the functions of these molecules? A little of the structures also. What else? So I guess we are done of biochemistry for your syllabus. According to your syllabus, I am finished with the biochemistry part. From next class onwards, I will be starting the molecular biology. Or maybe microbiology. We'll discuss with Professor Ghosh and then let you know what we'll have to start. Okay. And ma'am has given her portion for this thing, right? Your